Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphall, your host and guide through the fascinating future of the brain. Joining me today is Dr. Alysson Muotri, a professor in the departments of Pediatrics and Cellular and Molecular Medicine at the University of California, San Diego. Today, we will discuss how gaining a clearer understanding of the human brain evolution is critical for interpreting human genetic variants which lead to diseases. So welcome, Alysson.
Thank you so much, Deborah. Happy to be here.
So your research asks this really profound question: what makes us uniquely human? So what are the insights that you've gained by trying to answer that question?
That's a difficult question, and I'm still gaining insights on it. But I think there are a couple of things that most people agree. And I think most people agree that one of the hallmarks of our species is our ability to collaborate. This ability comes with the social brain. So we actually develop like the frontal lobe to be highly social. So this is the part of the brain that we use for cognition, but also to interact with other people. So we humans, we have this remarkable ability that is very different from other species. Even our closest relative, the chimpanzees, cannot cope with like a population that is as abundant, as higher as ours. So their colonies contains about 50 individuals. If the colony grows too much, there are a crisis. So they're going to fight. Something's going to happen. And we live in these large cities with millions of people, and we do just fine. And that's because we collaborate. We trust each other. We have the ability to live in larger cities. And this is due to this evolution that happens millions of years and shape the way we are now, especially our cortex, our brains.
So why this question? What got you onto this question? And what was that kind of inflection point for you?
I think that starts when I was a child, and I loved to be in nature and observe things. And I realized that, even the insects, social insects, such as bees or ants, they seem like very social, but at the end of the day, they are not. I mean, they are programmed to do that. The bee queen, you don't see like a revolution trying to cut the head of the queen. So we do this kind of stuff because we're always fighting for better quality of life. We are not like super happy with our state. We keep searching for something else. This searching for something else, that's what sparked my interest on figuring out what is unique about the human brain. So I decided to devote myself to study neurosciences.
So you studied neuroscience, but yet you're still looking at other species and trying to answer that. I mean, it's a big enough effort to just look at the human brain. How do you take in, the other information to be able to just be more broad in your perspective?
Yeah, that's a great question. And you realize that most of the neuroscience community actually use animal models. And we use animal models because of the inaccessibility of the human brain. We cannot, of course, open the skull and start studying the human brain, performing experiments to see how the brain will work. That would be unethical to people. So science find a way on dealing with animals and trying to extrapolate what we learn from animals to the human brain. So that was the part that I was, when I started to go in deep into my experiments, I was frustrated from the point of view of animal rights, as well as the type of insights that we were gaining by studying animals and trying to extrapolate to humans. So all of that motivates me to start looking for a better model.
That's really interesting. And you said some of these traits, especially around like being able to kind of collaborate, might make us more vulnerable to diseases. Is that true?
Yeah, so there is likely a trade off during evolution. We acquire a more sophisticated brain able to do those kind of things. But at the same time, it becomes more vulnerable. It's like having a very sophisticated car with all the electronics. So it's easy to break like a piece here and the car won't work, right? Whereas those like analogical machines, it's hard to break. So I think that's the same analogy. So over the course of evolution, we were gaining more and more abilities, but that make us vulnerable as well. And one of these vulnerability, it does affect the social brain, such as autism. Autism is one example of mutations, alterations in your genome that actually makes your brain less social.
Hmm. So your research, you've explored kind of ancient DNA, and you've gone all the way back to Neanderthals, and it's a pretty interesting kind of creative way to solve this problem. Can you explain that? And then, the second part here is, do you believe there was autism, thousands of years ago?
Yeah, both great questions. So the first thing is, how do we study the human brain? How do we understand the human brain? And as I mentioned before, all the tools that we have are not really at high resolution. We can study the brain in a non-invasive way. We can use imaging tools. We can use an EEG. We can use an MRI. But that doesn't give us what actually happens inside the brain at the level of synapses, at the molecular level. So I was looking for this kind of model. That's when I turned into stem cells. So we could use human pluripotent stem cells. Pluripotent means that they are able to specialize in different tissues of the body. And we're using those cells to actually create brain cells. We can even be more sophisticated. We can do this in 3 dimensions and create what we now call a brain organoid. So these are 3 dimensional tissues made of stem cells in the lab that mimics several aspects of the human brain. For example, it has all the major cell types, excitatory neurons, inhibitory neurons, glia cells. It forms circuitries. It forms some kind of anatomical structure that resemble the embryonic brain. It is not a perfect tool. It doesn't have vascularization. It doesn't grow that much. It's a tiny version of the tissue, but nonetheless, it's enough for us to start using this as a tool where we can manipulate in the lab, do whatever experiments we want without actually harm the actual human brain. So it becomes like an avatar of the brain. And because we can do that from live cells, from many people, we can choose people to study and see how their brains would evolve, how their brains will develop over time. So I actually been doing this, comparing like neurotypicals and people with genetic disorders to try to figure out what goes wrong. From the evolutionary perspective, I always wanted to do that with the Neanderthals. But unfortunately, we don't have live cells from the Neanderthals, so I cannot use their cells. But we do have the genomic information. So by comparing the genome of Neanderthals and modern humans, I can ask the question, what is unique about us? And surprisingly, we found that only 61 protein coding genes are different between us and the Neanderthals. And one of these genes, for example, is a master regulator of neurodevelopment. And if you CRISPR out, or let me put like in a more lay term, if you use genome editing tools to swap the modern gene for the Neanderthal version, we see that the tissue that is formed is very different from what we used to have. So it accelerates neurodevelopment. The brains mature faster. So this is one of the first results that we have by swapping Neanderthal genes inside human genomes.
So that's really interesting. So does that give us some idea of millions of years ago that the brain just didn't develop as fast, and now today, within the last hundreds of years, our brains are developing quicker? What would be the reason for that acceleration if that is true?
Yeah, so it's exactly the opposite.
Oh, okay.
So in the past, and even with the Neanderthals, we see an accelerated neurodevelopment. So the brains mature much faster. So we modern humans, we have exactly the opposite. We acquire these mutations that makes our brain develop dramatically slower compared to any other species, even the Neanderthals. So this is interesting because, for example, it seems like counterintuitive, but it's not. If you look into a baby chimpanzee, it can outsmart a human baby by far. So the a primate baby can already like find food, can be smart about the environment, but we have to nurture our little ones until they become independent. That takes a lot of energy and time. But if they grow up, they become smarter. I mean, they can actually outsmart the adult chimpanzee in some aspects, not everything. I mean, the animals are still the animals still do have some abilities that are even better than us. But overall, we acquired this sophisticated brain that is able to, again, social cooperation, imagination. We can transform our environment like any other species could do. So for some reason, yeah, we were selected to have a slower development throughout evolution.
So is that slower development? And thank you for that. That makes sense when you apply the chimpanzee and the babies. That makes sense. So is the slower development of our brains, does that open the door to mutations? Is that what's happening?
Yeah, you are absolutely right. The slow development of the brain allows for more things to go wrong. So you can imagine during the brain development, we have migrations of cells. They have to find the right circuitries. The neurons need to attach to each other. And then they start creating circuitries, and those circuitries will dictate behavior. So the faster you do it, the less opportunities you have for the system to mess up or for a mutation to occur. But we know we are giving plenty of chances for things to go wrong by having this very slow development. That's exactly the trade-off.
Yeah, that's so interesting. I know you study autism, you study other conditions of the brain. Why, if you would speak a little bit about why you have kind of delved into autism and epilepsy? You have a personal story that's really beautiful.
Right. And my personal story or my interest for this precedes my personal story. I was already studying autism. As a geneticist, I was looking for autism as a mutant where the brain, the social brain will not work. So looking into how the autism behaves, I could gain insights on how the neurotypical social brain works. So I was already studying that. And then I had a kid with autism. So my son, Ivan, he's 18 now, and he suffers from profound autism. So I choose two important words here. The first one is he suffers. The second one is profound. And I'm making this distinction because autism being a spectrum, there are several people under the spectrum of autism that doesn't suffer and doesn't have profound autism. So the work that I do are directly to this category, the ones that actually need medical help. And I'm saying that because sometimes I met some people with autism and they said, oh, I don't need a cure. I don't need a treatment. I'm fine. I have like remarkable abilities that you don't have. That is fine. But there are other kids on the spectrum that definitely need help. And one of the comorbidity that comes with profound autism is intractable epilepsy, meaning that you're going to have convulsions, seizures throughout life. So because of those things, I decided to make my research more translatable. So I stopped focusing too much on the academic side of it and start looking into ways where I can take the knowledge and translate into a medical benefit for individuals with autism.
So I appreciate that. Have you, through this kind of journey of your research and the personal, are we any closer to understanding why this profound autism occurs? Because you've talked about the evolution of the brain. Do we know why that happens?
Yeah. So on the past, I would say 10 years, we gain lots of insights on the genetic of autism. And this is because the cost of genome sequencing has dramatically lower, allowing us to perform more sequencing on people with autism. And what we are learning, still learning, but what we learn is that there is probably 1,000 genes or a little bit over 1,000 genes that regulates neurodevelopment that when mutated can lead to autism or syndromic forms of autism that can profoundly affect the person. And these are usually like point mutations that might be acquired throughout gestation. This is what we call de novo mutation, or might be heritable from the family and leads to autism or conditions related to the autism spectrum. So we know the genes, and we are understanding a couple of those genes, how those genes actually affect or leads to alterations in the networks and behavior. And we do, this is very specific experiments that we do in the lab to prove causation. So for a handful of those genes, we're pretty confident that we understand how they work, and there is a causal relationship between those mutations. And again, the fact that we have so many of those genes controlling neurodevelopment increases our chances as a species to have autism. You don't see autism in other animals, as far as I can tell. Even animals in the wild doesn't show signs of autism. Or even if they do, they will probably be selected out throughout evolution, and we don't see them. They will not survive alone in the wild. So that's why we think that autism is likely a human condition.
So you talked about stem cells, and you talked about these organelles, or kind of the organ in the dish or tissues in the dish. I think there's some people that still remember the conversation around stem cells, like in 1980, where there was a big debate whether it's ethical or not because it was coming from fetus tissue. And I like to try to bring this up because the science has gone way beyond that. Can you explain where these stem cells come from, and like how far the science has come is really amazing, and why it's so powerful?
Yeah, in the late 90s, early 2000s, we learned that we can make these different tissue cells from human embryonic stem cells. These are also pluripotent stem cells, but these cells are isolated from the human embryo. And there is a huge enthusiasm with those cells, because if you can make all these tissues, you can think about regenerative medicine, for example. You could create dopaminergic neurons and transplant it into the Parkinsonian brain to cure Parkinson. You could create motor neurons and transplant into someone who suffered from a spinal cord disease or trauma and make the person walk again. So everybody was super excited about regenerative medicine, and that would require the use of these embryonic stem cells. At the time, the only source that we had was the discarded embryo from in vitro fertilization clinics. So some of these couples will donate the embryos to science, and that's where we can get the stem cells from. And there was some groups of the society that were against the destruction of the embryos for science. And there was a huge ban on the use of embryonic stem cells in different countries, including US. But then, in 2006, Shinya Yamanaka, who later ended up winning the Nobel Prize in 2012 for medicine, discovered that you can actually reprogram cells from live people. So you don't need to destroy embryos anymore. I can take like a skin cell from you or a hair cell and take to the lab and activate some of these genes, and the cells will turn into these pluripotent stem cells that I could use to make whatever tissue I want. That solves two problems. It eliminates the ethical concern about the destruction of the embryos, and also capture the genome of the person. So if you are thinking about regenerative medicine, now I have cells from the same person that I reprogrammed from them. So eliminating the problem of tissue rejection later on. So that's the type of cells that we use. I actually reprogram cells from my research directly from the subjects. My son is one of the subjects of my research, for example. So I can study his brain without having to open his skull and performing experiments on him. I actually have his brain avatar in my lab where I can study. And I do that for several other neurological disorders as well.
What is that like to be able to, you see your son during the day and then, or nighttime, and then you go into the lab and you see his tissue under the microscope and it's doing things because the way this little, tissue in the petri dish there, you can actually see things moving, correct? You can actually see kind of that change that's happening. That's just really profound. Like, what does that feel like to you?
Yeah, it's a mixed feelings because, yeah, the tissue is alive. For example, you can test drugs to see if any of these drugs would make the neurons better. You can see that the neurons are struggling to make synapses or to form the right networks. So we can quantify those things. And you can, again, test potential therapeutics as well. So this is all very interesting and exciting. The part that makes me sad is that all these experiments takes a long time and they are super expensive. So to perform these experiments, it really requires like millions of dollars. So that's, as a father, I see how science goes slow because we don't have the proper funding for that. So I'm constantly struggling to support the next experiment.
Yeah. Yeah, which really, the thing that kind of I took away when we met a month or so ago was, you're really interested in the change that happens. We've moved from kind of a static, this is what the disease is, defining it under a microscope where it doesn't move and we try to hypothesize what's happening. You're really watching it either evolve or react or, you know, that is really that ability to do that is accelerating up. But yet, our systems, if you would, are still pretty slow in supporting the acceleration of science.
Yeah. No, you're right. And science hasn't changed the way we do it in over like 100 years. I think it's time for us to find like better ways to support science and really, remove ourselves from everything that keep us doing exactly the same thing. The current system do not reward, for example, high risk, high reward type of sciences because every investigator, every lab wants to have like some positive data so they can move from one grant to another. So that doesn't favor like trying different things. And it is a problem. So in the past, I would say that early investigators were way more exploratory and not super interested on incremental science. Now we see the opposite. Most people are doing incremental science and they are afraid to do like big jumps. But these are the big jumps that actually move the humanity forward. So we're not supporting them.
Yeah. Is that just communication? Is that communication to people that would be willing to take that risk? And I'm thinking outside the current kind of funding channels. Like maybe there's an opportunity here for, non-traditional investors that want to take that leap, maybe don't know science very much, but, wants to get involved. Is that a possibility? And how do we need to change how we get in contact with those people?
Yeah, no, I think that this is exactly what we should be doing. And we should interact more with people that are outside of the traditional financial support system. Like here in US, we have NIH, we have NSF, but these are agencies that are very traditional in the way they support science. But there are many people with lots of money out there who might be interested on supporting the science, but they might not have access or they don't know how it works or they never met like a scientist. So that's why this podcast is important because you take the word out and hopefully someone will see, will get interested, will figure out that, oh, this is an interesting topic. This guy is making brains in the lab. What does it mean, right? And might be interested on supporting the science. We need more outreach.
Yeah, I think that's really important because it's different tribes, if you would, or different different groups. They have different languages. They have different and they kind of stay within their own realm. And the real innovation comes when they, there's this outreach, or they start working together in kind of non-traditional ways. I think that's really important. One of the non-traditional things that you're doing, which I just find really incredible, is you put some of these brains in a dish up in space.
Yeah, yeah.
Tell me about that.
Right. Yeah. So once we create these brain organoids or brain tissue in a dish, the model is so good that it follows human development. And we start by saying that human development is so slow, right? I mean, it takes 9 months inside the uterus, and then the brain continues to mature. So one thing that I was really frustrated is that Ivan, my son, when he turns 9 years old, he start develop seizures, right? And I couldn't predict that. I already had like his organoids or his brain cells in my lab, and they never show up any signs of that. And most likely because they were not mature enough to show any signs of seizures. So I started looking for ways to accelerated maturation in the lab so I don't have to wait for 10, 20 years. And to the extreme, I mean, if I want to study Alzheimer's or dementia, do I need to wait for 70, 80 years? That's not practical. I won't be here when these cells were mature enough. So looking for ways to accelerated maturation, I came across some NASA literature where people have been analyzing cells from astronauts or experiments that were performed at the International Space Station. And upon return to Earth, they all show signs of senescence, meaning that the cell seems to be aging faster after exposure to space. So I thought that, well, I mean, if this is happening with blood cells, it might be happening with brain cells as well. So let's try. And that was our first mission in 2019, where we create these brain organoids from different people. And we send in a payload. It went up to the space station using like a SpaceX rocket. And it stayed there for about 30 days. It doesn't require any astronaut involvement. This was an automated payload that was keeping these brain cells alive. And after 30 days, they send it back to us. And when we receive the cells and we analyze them, and we can look into molecular clocks to measure the age of the cells. We saw that the cells have aged like 10 years or more. And we said, wow, 30 days in space make these brain cells becomes like 10-year-old. And I thought that, wow, that's a very interesting approach. And since then, we start sending more and more payloads to understand the basic fundamental aspects, why the space environment are aging the cells. And can we leverage the aging of the cells in space to actually accelerate drug discovery here on Earth? So this is the kind of science that we're doing now.
I would think NASA would be fascinated with that, because as we try to put people on the moon and then go into Mars, if you're putting 30-year-old, 35-year-olds, but yet they're now in space for a couple years, building a colony in Mars, there's a potential that their brain now is 65, 70 years old, that we just accelerated a disease that they may have gotten because they went to space.
Yeah. So I like to think that my research serves two purposes. It helps NASA to find neuroprotective agents for astronauts so they can actually go into long-term space flights or interplanetary colonization without like the damage of space. And at the same time, I'm leveraging this knowledge to help people on Earth. So actually the experiments that we do, can be used in both ways.
Yeah. You're an entrepreneur too, because your whole thing is trying to understand the change. You're trying to accelerate up some understanding. Tell us about that, because I think you have created a couple companies, if not more. Can you explain?
Yeah. I'm always like to be involved with startups, and most of them are spin-offs of the technology that we develop here in the lab, and always with the aim of making the science more accessible. So most of these companies have the aim to take the technology, improve them, and use so more people can benefit. So some of these companies are related to personalized medicine, others drug discovery, others to make better reagents for science in a cheaper way, in a more economical way, without use of animals. So all these companies have like some goals to really democratize science.
Yeah. And I got the opportunity to meet some of the people that are in your lab, and they're all very entrepreneurial. They're all very kind of excited, not just about the science, but what could be. How do you recognize that talent?
I think they are coming to me. So I'm not like doing like too much effort to select them. I mean, they reach out normally, and then we talk and we interview. And I think at this stage, I mean, they know my story, they know me, they see the type of science that I do, and they become very attractive. So yeah, I tend to select itself of people who have this entrepreneur mind, but also people who are not looking to just incremental science and it's willing to take the risk. So in the past, I would say that some of my students would like to become like an academic professor. And now none of them want. And I think that are a reflection of the times, but also the possibility, what you can do with the science and discoveries that you make in the lab, you can take to a next level.
Yeah, but you're creating a culture there that allows for them to be that, because some other labs might stamp that out, and that they wouldn't they wouldn't go that far. They wouldn't be taking those risks. What do you think you're doing that's creating that culture of excitement and what ifs and kind of challenging the norms?
Yeah. I think it's to guide by example, right? I mean, they see me, they see how I react, and I try to have them participate on discussions where we are either applying for a grant or creating a new company. And I help them all the process. So they see how I behave. They try to behave as well. When you visit the lab, I mean, there were like students very curious about, I mean, what they do, how do I approach, why do you approach me, things like that. And I think this kind of set the examples. But you are absolutely right to say that it's not for everyone. I used to put like an advertise for my lab that was inspired by one of these old adventure guys who was recruiting people. And I can send you the ad at the end. And he said, don't come here expecting that you're going to be rich or you're going to like strike in the first year. So here's exactly the opposite. The salary is low. Most likely going to be working off hours on the weekends, but it really requires some kind of a personality. So you have to be really passionate about that. And honestly, there were people who came to the lab and saw the culture and said, no, it's not for me. It's too risky, right? I'd rather follow like a more traditional pathway. And science is good for both. I mean, we need both people. We need some people more incremental too, because they validate the science, they add a little bit more and keep doing that. So it's important as well.
Well, you're very excited about what you're doing, and it definitely comes through. What, when you look out maybe 5-10 years, pick your horizon, what are you the most excited about when it comes to this kind of convergence of science and entrepreneurship?
Yeah, I hope at one point it will merge and would be hard to distinguish. I think we're going to be working together so well that would be space for exploratory science where scientists can actually follow whatever intuition they have. This is something that we no longer even encourage people to do it. On the other hand, there is going to be like translational applications where you can use the technology that you discover to really apply to a better world. So my hope and my dream is that like in 5-10 years, this would be closer and closer to each other and almost indistinguishable. The same group of people will have individuals with those two personalities and they get along and they don't have to be in either a company or an academic setting. It's going to be merged.
Yeah. So if an investor with a lot of money came walking through your lab door and said, what are the 2 or 3 biggest gaps that we should be funding for the future of humanity? What would you say?
I would say that I would invest the funds on improving the technology that we have, but not like incremental improvement. I think we need like really non-traditional ways of doing things. For example, I want to create like a brain organoid that is no longer an organoid, but an actual human brain in the lab. So we don't have to to have limitations. We can reproduce the human brain in the lab. And I think it's possible to do that if we have the right funding. The other thing that I would do if I have unlimited funding would be to create like the first stem cell lab in space, so we can manufacture brain tissue for drug discovery, accelerated drug discovery, taking advantage of the space aging process.
Yeah. Yeah. Wouldn't that, maybe there's somebody out there that wants to do that, hopefully. Hopefully. So again, thinking about the future, thinking about your career, you're a young man. What do you want to be kind of looking back 15, 20 years? What do you hope, that you can be able to say, boy, I did well, or what do you want to be known for?
I try not to think about that because I think like legacy means nothing. We all gonna die and people will forget everything. I don't even remember who won the Nobel Prize last year. So why do I care? I don't care about that, right? So I try not to think. I think about today, what I can contribute today. And I think my contribution today, it is on mentoring next generations, right? I mean, see if I can make them wake up for a new way of doing science. The other thing that I'm highly involved, it is to incorporate more traditional medicine into modern science. And I'm doing that with the original tribes of the Amazon, for example, where we talk to the shamans and look for this ancestral knowledge. So they're pointing us to some plants or some molecules that are neuroactive and could be neuroprotective. So who knows, maybe the cure for Alzheimer's or Parkinson would be in one of these plants. So I kind of believe that the nature has lots of resources, and we are not exploring that in a good way and ignoring the traditional medicine that has been there for thousands of years doesn't help. So I want to reestablish the dialogue between ancestral knowledge and modern science.
Yeah, that's really interesting. Which brings up a whole bunch of questions about the traditional way that we get drugs through. But, if we can find that, maybe we can find a way to get things approved too. As we kind of wrap up here, what is the question I should have been asking you? Because you've got your hands in everything. What is the question that you wish I would have asked or should have asked about the importance of what you're doing?
Yeah, I think there is one important question that you could have asked me. It is, for example, if we are to take these brain tissues created in the lab to the next level, would they acquire self-conscious? Would they become self-conscious? Would they have some kind of a consciousness that we should be aware? Are those entities that we create in the lab, do they deserve some kind of personal rights? The same way that we should treat like a chimpanzee as an individual, should we treat this brain creates in the lab as an individual as well? And now, I think I mentioned to you some of the research here is incorporating this brain tissue into machines. And we actually have cyborgs walking around and they are learning how to control their movements, not using a computer, but using a brain tissue that we create in the lab. So what if they start acquiring some kind of consciousness? Is it unethical to do it? Who owns them? The person who donates the cells? So there's lots of ethical questions that I think in the next few years, we should discuss them more, because I think the way we are going is become inevitable for us to create like a human brain with some level of consciousness fully developed on them. And I don't think we are prepared to answer those questions yet, but the science won't wait. It will happen. And I like to, it's not like an alarm, but I like to start those discussions because I remember when Dolly, the clone, happens. And I think most people were taken by surprise. They didn't know that science was so advanced to be able to clone a mammal. And I remember all those discussions and people thinking, oh, now the next stage, they're going to do it with a human. So we can prevent those kind of alarmist discussion if we start discussing it now. So I think that's one important.
With you, we need to start asking that question. So, Alysson, if people have this great big bag of money that they want to give to you to start the stem cell lab up in space, how do people get a hold of you if they're interested in what you're doing or want to fund you or just learn more? How do people get in contact with you?
Yeah, all my information is in the internet. It's very easy to find the lab page for my lab. You just Google my name, you're going to find it. The best way is by email. And again, my email is available.
Wonderful. I really appreciate what you do. And I really appreciate the non-traditional looks that you're giving science. And it's important to all of us. And thank you so much for your time today.
Thanks, Deborah. Awesome questions. Thank you so much.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.